Today I will talk about the DNA fingerprinting. DNA fingerprinting is a forensic technique used to identify individuals by characteristics of their DNA. The technique of DNA fingerprinting or DNA profiling was developed by Dr. Alex Jeffrey. DNA fingerprinting involves identifying difference in some specific region in DNA sequence called as repetitive DNA, because in these sequences a small stretch of DNA is repetitive many times. At this point, we shall go over a few key concepts. Centromeric satellite DNA. This type of satellite DNA is found at the centromere, which are essential for chromosome segregation during cell division. Centromeric satellite DNA sequences are highly repetitive and play a crucial role in the formation of the kinetochores. Other is the telomeric satellite DNA. Telomeres are located at the end of the chromosomes and are involved in the maintaining genomic stability and protecting the integrity of the genetic material. Telomeric satellite DNA consists of a repetitive sequence that form the protective caps at the chromosome ends. Moderately repetitive sequences, which contains 150 to 300 base pairs. They include transposon. On the other hand, mini satellite sequences, they are 11 to 60 base pair long, sometimes 10 to 100 base pair. They are VNTR, or means variable number of tandem repeats, and used in DNA fingerprinting. Other is microsatellite sequences. They have 5 to 8 base pairs. Microsatellite or simple sequence repeats SSR occur in genome over a million copies. These repeated sequences are called junk DNA. So what are the features of the satellite DNA? Number one, satellite DNA produces a pattern of bank that is unique for every individual. VNTR vary in number from person to person. These sequences normally don't code for any protein. They show high degree of polymorphism in population. They are inheritable from parent to children. So what are the types of DNA fingerprinting? There are 4 types of DNA fingerprinting. One is RFLP, means restriction fragment length polymorphism. It is one of the earliest method. It requires a large amount of high quality DNA. Second is PCR based fingerprinting. PCR means polymerase chain reaction. Third is RAPD, random amplified polymorphic DNA. It is quick but less reproducible. And fourth is AFLP, amplified fragment length polymorphism. Now we see the technique of DNA fingerprinting. DNA fingerprinting uses restriction fragment length polymorphism, RFLP. analysis techniques to analyze VNTR. It includes number 1, isolation of DNA. Number 2, digestion of DNA by restriction endonuclease. Number 3, separation of DNA fragments by electrophoresis. Number 4, transferring means blotting of separated DNA fragments to synthetic membrane such as nitrocellulose or nylon. And 5th, southern blot hybridization using labeled VNTR probe. And 6, detection of hybridized DNA fragment by autoradiography. 7th is DNA amplification is done by PCR, polymerase chain reaction. What are the steps involved in DNA fingerprinting? Number 1 step, isolation of DNA. The DNA can be isolated even from the small amount of tissue like blood, hair, roots, skin, etc. Number 2 is restriction digestion. The isolated DNA is treated with restriction enzymes which cut the DNA at specific site to form small fragments of variable length. Variation in the length of restriction fragments are known as restriction fragment length polymorphism, RFLP. Number 3 is gel electrophoresis. The DNA sample are loaded on agar gel and electrophoresis is carried out. Negatively charged DNA fragments move to the positive pole. Separation of fragments depends on their length and it results in the formation of band. Double-stranded DNA is then denatured into single-stranded DNA by alkali treatment. Number 4 is southern blotting. The separated DNA fragments are transferred to a nylon membrane or a nitrocellulose membrane. Number 5th, selection of DNA probe. DNA probe is known sequence of a single-stranded DNA. It is obtained from organisms or prepared by cDNA preparation method. The DNA probe is labeled with a radio-labeled isotope, and the 6th is hybridization. In this process, probe is added to the nitrocellulose membrane containing DNA fragments. The single-stranded DNA probe pairs with the complementary base sequences of the DNA strand. As a result, DNA-DNA hybrids are formed on the nitrocellulose membrane. Unbound single-stranded DNA probe fragments are washed off. 7 is photography. The nitrocellulose membrane is then kept in contact with the X-ray film. DNA bands due to a radioactive probe give photographic image on X-ray film. This is autoradiography. Now we see the application of DNA fingerprinting. Number 1, DNA fingerprinting useful in crime investigation in forensic sciences. Number 2, matching organ donor to people needing an organ replacement. Number 3, diagnosis inherited disorder in parental and newborn babies. Research on genetic disorder. Number 4. Number 5th is it can be used to determine whether a particular person is the parent of a child. Means child paternity and maternity can be determined. At this point, we shall go over a few key concepts. Number one is DNA polymorphism. The presence of two or more variant forms of specific DNA sequences that can be occur among different individuals or population. If more than one allele, such as ABO blood group, occurs with a frequency greater than 0.01, it is called DNA polymorphism. Probability of such variation in non-coding DNA sequence is more. For every evolution and speciation, such polymorphism play an important role. So what are the causes of DNA polymorphism? DNA polymorphism can be introduced into a population by mutation and genetic drift. Types of DNA polymorphism. One is RFLP, means restriction fragment length polymorphism. Number two is VNTR, variable number tandem repeat. Number three, SSR or STR, simple sequence repeat or short tandem repeat. Number four, SNP, single nucleotide polymorphism of DNA polymorphism. So what is autoradiography? Auto means self, radiogram equal to negative photographic image of an object produced on the film with rays emitted from a radiation source away from the object. Autoradiography allows the visualizations of molecules or fragments of molecule marked with radioactive elements. The autoradiogram will gives bands of same size in monozygotic twin. The sensitivity of this technique has been increased by use of PCR. Now we see the technique of blotting. Blotting is a technique by which a macromolecule such as DNA or RNA or protein is resolved in a gel matrix, transferred to a solid support and detected with a specific probe. Blotting is a technique in which nucleic acid such as RNA and DNA or protein are transferred onto a specific membrane. This membrane may be nitrocellulose, PVDF or nylon membrane. This process can be done just after the gel electrophoresis by transferring the molecule from the gel onto the surface of the blotting membrane. Southern blotting is used for DNA analysis, Western blotting is for protein analysis, Northern blotting is for RNA analysis, and Eastern for carbohydrate detection.